Bismillah ar-Rahman ar-Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. In this video lecture, we are going to talk about gram-positive cell wall. Okay. Now we already talked about the peptidoglycan structure and how the peptidoglycan is actually synthesized, and we already talked about the bacterial cell wall overview. Now this is the third video. This is the third part of the bacterial cell wall regarded with the cell, and in this third part, what we are going to do, we are going to talk about the gram-positive cell wall. Gram-positive cell wall. Gram-positive cell wall is actually thick, and it is contains 70 to 100 layer. Okay, it contains 70 to 100 layer of the peptidoglycan. If you don't know about the peptidoglycan, so go back to my channel and watch the video about the peptidoglycan structure and synthesis. Okay, so it contains 70 to 100 peptidoglycan. How? Let me draw here. So first of all, this is the first and this is the second peptidoglycan layer. This is the third, fourth, and then we have again these are what? These are actually peptidoglycan. Now this black one are the name, and this is the neg. This red one are what? These are the neg. Again name and neg. Okay, so this is the layer of the peptidoglycan. What? This is the peptidoglycan, and it will be 70 to 100 layer of the peptidoglycan in case of the gram-positive bacteria. Now if you see, so this name, now this is the name. Okay, this black one are represent what? This is name, and this red one are what? This red one are the neg. So these two names will be connected with a peptide linkage, as I already discussed. So these are what? These are the peptide linkage. As you can see, these are the peptide linkage. Okay, with this peptide linkage, we have a structure like that. Let me draw with the blue marker. This structure are called what? This structure are called teichoic acid. Teichoic acid. After that, what we have? We have the cell membrane. Now this is the lipid layer, and this is the hydrophobic layer, and this is the hydrophilic layer. Okay, so this is the cell membrane. Sometimes this teichoic acid are also means extended from this cell membrane as well. Okay, which is connected with the lipid layer, and then we called it lipoteichoic acid. What we called it? We called it lipoteichoic acid. When it is connected with the lipid, so we call it lipoteichoic acid. This teichoic acid means if we see, this teichoic acid are actually positive in nature, so they are positively charged, means they are positively in nature, and they can attract negative substance which is present outside the cell. Now the function of this teichoic acid are unclear, but in some books they says that this teichoic acid are actually responsible to maintain the structure of the peptidoglycan layer, or we can say it can maintain the peptidoglycan structure. Now this teichoic acid is, as you can see, it is connected with this amino acid. So it is connected with this bond, means where the amino acid are connected. So see, this peptide bond, it is connected. Now, so how this teichoic acid will be look like? If we see the teichoic acid. So teichoic acid are actually, it consists, this teichoic acid are actually the polymer of what? This teichoic acid are actually the polymer of glycerol, which is connected with a phosphate group. So how it will be look like? It will be look like means there is a phosphate group and there will be the glycerol. And then again phosphate group, and then again means this CH2, and again carbon, and this alkyl group, and again CH2, again phosphate group, and then again hydrogen group. So this is actually the teichoic acid, means the teichoic acid are actually consist of phosphate group, as you can see, phosphate group. So it is connected with the phosphate group. Now there is a space, as you can see, this one space we called it periplasm. There are some proteins are filled, as I draw here with the, let's suppose I draw here with the green marker. These are the proteins, and this space is actually filled with this green proteins, and we call that exoenzyme. Enzymes, or we call it exoproteins. These exoproteins are actually responsible to degrade the large polymeric substance which are actually transmitted cell to cell. Okay, so that's why we call it periplasm. We not call that periplasmic space, because the space is actually occupied by this protein, and the space is actually occupied by this peptidoglycan, thickness of the peptidoglycan layer. That's why we call that periplasm, not periplasmic space. Okay, so this is about the gram-positive cell wall. Inshallah, in the next video lecture, we are going to talk about gram-negative cell wall. So please stay tuned and continue to watch the video. Thank you so much.